All right, welcome back. So here we're going to look at, we're going to pick up now talking about the meninges of the brain, and we're also going to look at the ventricular system of the brain as well. So with these sheep brains, we unfortunately do not have the meninges still attached. There are no meninges on these sheep brains, but what we can do then is we can talk about their location of where they would be if they were actually on the brain. So if you recall, there are three meninges, three meningeal layers called the dura, the arachnoid, and the pia mater, right? Dura, arachnoid, and pia. Dura means tough. It's the outermost, most superficial layer. You have the arachnoid, which means web-like or spider-like, right? So think of like arachnophobia. And then pia mater is the deepest, it's the innermost, and pia means delicate, right? So it's the thinnest, most delicate layer among the three meninges. So if we look at our brain, and again, this could be if we're looking at a sheep brain, this could be if we're looking at a human brain, we can look now and see where they would be on the brain, right? So if we take our sheep brain, right? If we're looking on the actual surface of the cerebrum, right? Actually on the surface, attached to the cerebrum, we would have the pia mater, right? The pia mater. The pia mater, you cannot, okay, hear me, you cannot take the pia and peel it off, right? It is firmly adhered, it is firmly attached to the cerebrum, right? And these layers that we're talking about, they would also exist around the spinal cord as well, right? So that's the deepest layer, right? The innermost attached to the brain's pia. Just superficial to that, you'd have the arachnoid mater, right? Arachnoid mater. And then superficial to that, the outermost, you would have the dura, right? The dura mater. The dura is the outermost, it's the toughest. You can actually cut it and remove it, and that is what has been done on these brains here, so they're not present, right? Now, relative to these meninges, there are spaces. Okay, so there's spaces superficial to, there are spaces deep to these meninges, right? And you may have heard of some of these. If you've ever heard of an epidural hematoma, or a subdural hematoma, or the subarachnoid space. All of these are spaces which may or may not have things in them, but these are spaces relative to these meninges. So one space that we want to look at, or that we want to concern ourselves with, is something called the subarachnoid space, right? The subarachnoid space. And I'm going to leave it there, and I'm going to come back to the subarachnoid space in just a second. But before we do that, we need to talk about something called the ventricles, or the ventricular system of the brain, right? So I'm going to show you the location of the ventricles on our sheep brain and our human brain, and then I'm going to show you a model of the ventricles, which may help it make a little bit more sense, right? So what these ventricles are, they are holes, they are cavities in our brain, essentially, right? There are these cavities, these openings in our brain. So if we look here at this sagittally sectioned brain, hopefully you can see, if we kind of look here toward the middle, right? If we're kind of looking in this inner region, you notice this big cavity, right? You can kind of stick the pointer down in there. You can also stick your finger deep in here, right? It's this deep cavity way in here, right? You can see all in here, that deep cavity. This is one of our ventricles, right? This is one of our ventricles, and we have four of them, right? We have four ventricles. We have two lateral, okay? So a lateral here, so this is one lateral, so lateral. The other lateral would be in the other cerebral hemisphere, right? But we've removed it, right? So there's one lateral over here, and there'd be another lateral on this side, but it's gone, right? So two lateral. You have what's called a third ventricle, which typically sits here in this region called the diencephalon, right? It usually sits in this general region, kind of in here, called the diencephalon. So the third ventricle is here. And then there's a duct that extends down, and the fourth ventricle is usually just anterior to the cerebellum. So kind of in this region, kind of all in this region here. This is the location where we would have our fourth ventricle, right? So lateral ventricles, location of third, approximate location of fourth, kind of all in this area right down in here, okay? So from our lateral, you have two lateral ventricles. The cerebral spinal fluid is what is in these. So the reason we're talking about these is there is a fluid called CSF, cerebrospinal fluid, right? And it exists in these ventricles, right? It is made in these ventricles in a tissue called the choroid plexus, which is essentially It's derived from the pia mater, right? But it's called the choroid plexus, and it makes CSF, makes cerebral spinal fluid. And this CSF is ultimately functioning to nourish, to cushion, and to provide some buoyancy to the brain. It also helps with some exchange of ions and other materials, right? But you have to go from lateral through an opening called the interventricular foramen, which is a little opening. So if you think of the word, interventricular means between the ventricle, and foramen means a hole. So interventricular foramen is a hole between the ventricle, right? And so that allows CSF to go from the 2 lateral ventricles to the third, which again, remember, is in the diencephalon, which again is this region here. The thalamus is also part of it here. From the third ventricle, there is a slender tube, the slender opening called the cerebral aqueduct, which extends from the third, and extends down, and it passes kind of posterior to the brainstem, and it drains CSF into our fourth ventricle here. And then from the fourth ventricle, there are what are called apertures, a median and 2 lateral apertures, which extend medially and laterally to allow CSF to then enter into the subarachnoid space, right? Which again, I mentioned a second ago and said we'd come back to it. So this CSF that circulates through these ventricles, it ultimately enters into the subarachnoid space, right? And that subarachnoid space exists around the brain, and it also exists around the spinal cord as well, right? So if we're looking at our human brain, the approximate location of some of those, we'd have our third ventricle kind of up in here, right, deep in the cerebrum. We'd have our lateral ventricles. Our lateral ventricles would be up here. We'd have our third ventricle in this region of our diencephalon. We'd have this cerebral aqueduct, which you can kind of trace as this little tube extending down. And then you'd have this fourth ventricle here, right? So lateral, third ventricle, fourth ventricle, and then from there, the CSF would enter into the subarachnoid space, right? So it may be kind of hard to conceptualize and to imagine these ventricles, but if I show you a model of the ventricles, it may make a little bit more sense. So this is a cast, a model of our ventricles, okay? And so it kind of looks abstract, it kind of looks interesting at first, but imagine for a second, you have your ventricles all here. Imagine you injected or inserted some type of foam or some other type of material into these ventricles, and then it hardened, and then you could peel it out, right? So like you're making a mold of something. That's essentially what this is, right? So these are our lateral ventricles, right? So this large one here, and it kind of makes this C shape here. So there's one on one side in one hemisphere, and another in our other hemisphere. So again, these are our lateral ventricles. Importantly, don't call these ventricles ventricle 1 and ventricle 2. Okay, just call them lateral. Okay, lateral ventricle, right? We said from our lateral ventricle, we go to our third, which is this one here with this opening here. This is our third ventricle, and to get there, you go through this little opening called the interventricular foramen, which is this little opening here. Kind of see it here. That allows the CSF to go from lateral to the third ventricle. From the third, we go down to our fourth, but we have to go through this long tube called the cerebral aqueduct, right? And this is our cerebral aqueduct, all right here. So that tube that would be extending down from our third all the way down into our fourth, that's the cerebral aqueduct, and so that's what we see. That's what we see right here, this long tube here. And then we go down to our fourth, and then from the fourth, we said that there are apertures, and these apertures extend into the subarachnoid space, and so you can see some of those apertures here, right? You can see these 2 lateral here, and then there'd be this medial here that extend out into the subarachnoid space, right? So again, lateral, just as a review, lateral, third, fourth, interventricular foramen, cerebral aqueduct, right? So that's the pathway, and then from there into the subarachnoid space. That's the pathway, the flow of CSF, of cerebral spinal fluid. Okay, so keep that in mind. It may be helpful to write out a progression. So we start in our lateral ventricles and we go to our interventricular foramen, to our third, to the cerebral aqueduct, to the fourth, to the apertures, to the subarachnoid space, and around, right? Now, I mentioned that you have this subarachnoid space around the brain and around the spinal cord. In the brain, however, we have these little extensions called arachnoid villi, or arachnoid granulations, which are extensions from the subarachnoid space that allow the CSF to be drained ultimately back into the blood. Okay, so the CSF comes from the blood and it's ultimately returned back to the blood. Okay, and these are called arachnoid granulations. Sometimes they're called arachnoid villi. At any one time around our brain, around our spinal cord, we may have about 150 milliliters of CSF circulating kind of through the ventricles and in the subarachnoid space. But in any given day, we may produce around 500 milliliters, okay? And so what this means is as we are producing CSF, right, in the choroid plexus, in these lateral and third ventricles is where you have the choroid plexus that makes the CSF. As you're making it, you're also having to reabsorb some of it as well, right? So you're having to collect some of that CSF back into the blood, and it largely goes back into some of the veins of the brain called venous sinuses is where the CSF is ultimately drained back into, right? So this is just a little review over some of our ventricles and our subarachnoid space and CSF, and we will pick up in our next video talking about some more cerebral structures.